Good morning, everyone. Today we have Professor Umut Gürkan from Case Western giving a seminar at the Terasaki seminar series. A little bit bio about Umut. He's a Warren Rupp Associate Professor in Case Western University in Mechanical and Aerospace Engineering Department. He also has a secondary appointment in the Department of Biomedical Engineering. He has a bachelor's in Chemical and Mechanical Engineering from Middle East Technical University, PhD from Biomedical Engineering in Purdue. He did a postdoctoral training in Harvard MIT Health Science and Technology. His honors awards include NSF Career Award, USPTO Patents Award, Biomedical Engineering Society Rising Award, has numerous awards. And he has 80 papers, numerous book chapters, numerous patents. He's super successful over there. We're happy to have him today. Umut, please go ahead.
Thank you, Mehmet, and thank you, Ali, and everyone at Terasaki Institute for having me today. It is one of the few virtual seminars that I have been invited to give over the, since the pandemic happened. So let's see how that goes. I still don't feel like I'm an expert in navigating these virtual seminars. I will try to keep it fun and entertaining, and also I'll try to keep the technical content a little bit, less intense, considering that it is still 8 a.m. over there. It is almost noon here in Cleveland. So I will talk about inherited disorders of hemoglobin. It's one of the most important proteins that we have in our bodies. That's the red protein that gives the blood its red color, as you all know. And I'm going to talk about some of the new developments and how our research relates to some of the new recent developments in this area, from diagnostic to cure. It's really exciting times for many diseases, especially diseases that are caused by a single genetic mutation, which is the case for many inherited hemoglobin disorders. And in sub-Saharan Africa, outside US, in sub-Saharan Africa and India, in low resource settings, typically, for example, in the picture that you see here, you see 3 kids living in a rural area in Ghana. That's a picture that was taken when I was visiting one of the villages in Ghana. And there's a chance that one of these 3 children is likely living with some kind of an inherited hemoglobin disorder. It can be heterozygous or homozygous. It can be sickle cell disease or hemoglobin C, which I'm going to talk about. So the prevalence is actually quite high in those regions. So our research has been quite translational since I started here at CASE, and these are some of the companies that licensed our technologies, going from right to left. DX now licensed some of our technologies from my postdoc work. This is a company with Utkan, whom you might be familiar with. The other 3 companies are originated from CASE. These are all focused on blood and developing diagnostic technologies for blood-based diseases. The clot chip Exatec is an hemostasis clot bleeding disorder focused company developing diagnostics for that purpose. Recently, this company received an US FDA breakthrough device designation. So it is pretty close to becoming a clinically applicable product. A new company that we are just starting this summer is BioChip Labs for customized rheological assays for red cell disorders. I'm going to briefly talk about that, but most of my presentation, actually maybe half of my presentation today, will be focusing on diagnostics for hemoglobin diseases based on a product that we licensed in that space. The product is called Gazelle. It's an integrated diagnostic system for hemoglobin variants, anemia, and malaria. It is commercialized by Hemex Health, and Biorad is one of the partners in this commercialization effort, and I will talk about it as well. This technology, by the way, received patents for humanity recognition. It has CE mark in Europe. The product launch has happened, so this is now a commercially available product outside US. So So learning objectives for today's presentation is, I'm actually realizing that when I give these talks, not many people know about the different types of hemoglobins that we might see in certain situations, and the inherited versions of these hemoglobin disorders, and how it might actually affect human health, and what are some of the emerging global trends in the treatment and the cure of these hemoglobin disorders. And in that context also, how can we rethink the diagnosis and monitoring of these disorders with a global perspective. So before I go into the hemoglobin, I would like to briefly talk about human circulation, especially microcirculation. These hemoglobin disorders, inherited hemoglobin disorders, typically has the biggest impact. They have the biggest impact on microcirculation. From a tissue engineering regenerative medicine perspective, as you are all aware most probably, microcirculation or microscale flow is quite important so that the tissues are oxygenated and they can stay alive, and the blood flow in microscale actually allow that. So if you have issues in the microcirculation in the body, which can be caused by the main component of blood, which are the red blood cells, and that becomes a big problem. Most of our research, most of our fundamental research in our lab focuses on understanding the relationship between red blood cell health and microcirculatory conditions. So how do the changes in red blood cell might cause problems in the microcirculation and enhance more systemic effects. Red blood cell health, which is also inherently related to the hemoglobin type in it, which I'm going to talk about later, can be associated with many different diseases. You can actually find publications and you can actually find clinical evidence suggesting that red blood cell health either deteriorates, or a deterioration in red blood cell health can cause all these diseases that you see here. At the top of the pyramid, which are the most severe versions of red blood cell disorders, are severe anemias. Sickle cell anemia is being one of the most well-known. Below that, infectious diseases such as malaria and sepsis can cause changes in red blood cells, or the red blood cell changes can make things worse in these conditions. Toxic elements such as environmental toxins, mercury, lead have impact on the red blood cell, which can again go and change things in the microcirculation in a negative way. If you are dependent on blood transfusions or bank blood, that might have impact on the microcirculation, because the bank blood quality is not as good as the fresh red blood cells that are produced in the body. Diabetes can have an association with red blood cell health and vice versa. And kidney diseases, certain kidney diseases, actually, especially end-stage renal disease, has been associated with red blood cell health and microcirculatory problems, especially if you're on dialysis. That is thought to have impact on the red cells and the microcirculation. And the other one is the cardiovascular diseases. There's actually a close relationship between the two. So we have done work, and we have been doing work in many of these areas, but primarily our focus is at the top of the pyramid, the most severe versions of microcirculatory and red blood cell health. Severe anemias, malaria being the top two conditions that we work on. So as I said, we did some work in other areas. For example, this is a tweet from Francis Collins a few years ago. He highlighted in his blog our work on microfluidic assessment of red blood cell health when these red blood cells are exposed to mercury. So we looked at how mercury as a toxin might change red blood cell quality, and which might have implications in microcirculatory health, endothelial function, and so on. And we also use scientific illustration in collaboration with Cleveland Institute of Art. We have an excellent art institute here, and they have a biomedical art program. So we work with them closely, illustrating and basically trying to better disseminate our research findings and our technologies to the general public. And it's an indication of This slide actually is an indication of how art can actually help us, better explain our science and for an increased public interest. So let me show you an animation of what happens to the microcirculation if you have unhealthy red blood cells or if you have an abnormal hemoglobin. In this case, this is a representation of sickle cell disease. If the red blood cell is unhealthy and if it has a variant of hemoglobin, sickle hemoglobin in this case, it can deform. In this case, it is represented as a sickle shape. That's why this disease is called sickle cell disease. These mutations are typically a single mutation that is, for example, in this case, a T mutation in the gene that is responsible for producing hemoglobin. And this causes a slightly different version of the hemoglobin that's highlighted by this green single amino acid difference. This actually causes the hemoglobin to polymerize in low oxygen conditions, actually makes the hemoglobin a little bit hydrophobic. That helps the hemoglobin polymerize in certain conditions in the red blood cells. And when the polymers form, the hemoglobin polymers form inside the cell, that causes the cells to deform, changes the membrane properties, increases the stiffness, and so on. And all of a sudden, it becomes a rheological problem. So there are many different types of hemoglobin disorders. Actually, there are more than 1,000 different types of mutations that have been identified. The top three being hemoglobin S, hemoglobin C, and hemoglobin E mutations. S mutation is common in sub-Saharan Africa. C is common in West Sub-Saharan Africa, where Ghana is, and west of Ghana especially. Hemoglobin E is common in India and Southeast Asia. So we have worked with all these three types of hemoglobin variants so far. We developed diagnostic tests for them, which I'm going to talk about. We also have done some work at trying to understand how the combinations of these mutations or how the homozygous versions of these mutations change red blood cell health and microcirculatory health. As in many different types of, especially recessive genetic disorders, you might actually have three different conditions or three different possibilities. AA, or the normal condition, healthy condition, is the condition where you have two copies of the healthy hemoglobin gene from your parents. The trait or heterozygous state is in which you have a healthy and you have an unhealthy copy of the gene from your parents. And the SS is the homozygous version in which you have inherited two copies or both copies of the mutated gene from your parents. So homozygous state is the one that is typically associated with these problems that we typically focus on. So you might be wondering, how these mutations emerge and how they survive over the generations. And if it is a disadvantage to have this mutation, then why do they exist? And why are there so many different types of hemoglobin mutations? These mutations are actually linked to malaria. So the mutation in the hemoglobin gene gives protection to the especially to the carrier. So if you have a mutation, for example, in hemoglobin, and if you inherit AS, if you are heterozygous for S, then you are protected against malaria. There is a misconception in the research community that SS might be giving a better protection against malaria, but that's not true. Subjects with SS are slightly less likely to get malaria, but if they get malaria, they are more likely to die because of it. So this advantage is actually basically benefited by the heterozygous state, AS group, so the middle group. So that's why these mutations actually exist and survive for many generations. And you might actually wonder why we have these different types of hemoglobin variants or mutations all over the world. So that's the probability, most likely, in different places, different mutations emerged and sustained as a response to malaria. And one of them, the sickle mutation, turned out to be the most severe version of these hemoglobins that causes that polymerization of hemoglobin. So as I said, malaria and these hemoglobin mutations are related, and the prevalence of malaria and hemoglobin variants are also, they coexist. So if a malaria is a prevalent disease in a certain region, it's quite likely that you will see a high prevalence of hemoglobin variants, especially sickle cell disease if it is Africa. And due to both of these conditions, you would expect to see more than 1,000 deaths daily due to malaria and sickle cell disease if you combine the global impact. So in the low resource settings, the key unmet need is the affordable diagnostics for early detection, and for hemoglobin variants, it is again early screening and diagnosis and management. And diagnostic needs and the importance of early diagnostics became quite obvious in the ongoing pandemic, as we all know, and this unmet need has been persisting for many years for these two conditions. And the current diagnostics are too expensive, inaccessible, require experts. The turnaround times are long. So here, for example, we observed that the COVID turnaround times for diagnostics can be 2 to 3 days in certain regions, which is unacceptable. If you go to Africa, for example, the turnaround time for a sickle cell diagnostic might be up to 2 to 3 weeks. So which is a really long time, and sometimes no diagnostics are available in the country. Samples should be shipped outside the country in certain regions, so that's really unacceptable. So both of these conditions, anemia, the sickle cell anemia and malaria, cause anemia. So it is important to look at the hemoglobin level in blood and monitor hemoglobin level in blood if you want to monitor the treatment of these conditions. The commonality between the two, one of the commonalities is anemia. So that's why it is important to look at the anemia state of these patients. And if you have a diagnostic approach, it's important to keep the anemia hemoglobin level in mind. So another important consideration is the prevalence and where these conditions are prevalent. In this case, more than 99% of people living with these conditions are living outside US in low and middle income countries, and many of these patients are in low resource settings. So the order of the slides that I'm actually presenting and the information that I'm providing is intentional. So I'm actually trying to provide the background so that we understand the problem better, we understand the regions that these problems exist better, so that when we come up with solutions as engineers, we keep these constraints in mind. So this is like the translational way of thinking based on our experiences. So another thing that I would like to look at the history of these conditions and what has been done and what has worked, what has not worked. If you compare the history of sickle cell disease, for example, in the US versus in Africa, you will see that about 60 years ago or so, the life expectancy in Africa and life expectancy in the US were more or less the same, which was less than 10 years of age. So average life expectancy was less than 10 years, like 60 years ago in the US, if you are born with homozygous version of the sickle cell disease. But over the years, things changed in the US for the better. With the National Sickle Cell Act, newborn screening has been mandatory. So everybody who is born in the United States are screened for sickle cell disease, regardless of the ethnicity, regardless of the origins. And everybody, all the babies are screened. So that's a requirement here. So that has helped with the early detection of the disease and management, which helped increase the life expectancy. In addition to that, certain treatments became available. Penicillin, which protects against infections. So these kids, when they are diagnosed with sickle cell disease, they are put on daily penicillin, and also, regular penicillin, and also daily hydroxyurea. Hydroxyurea is a drug that increases that changes the hemoglobin composition for the better, which improves the red blood cell properties, which I will briefly talk about later, and also transfusions. It is important to note that up until 2019, FDA did not approve any drugs that were specifically designed and developed for sickle cell disease. For these diseases, the approach has always been to repurpose existing drugs that were originally developed for other diseases. Cancer, for example, hydroxyurea is a cancer drug, and it was repurposed for sickle cell disease. So the development in the pharmaceutical area for this disease has been quite slow, and but I will briefly talk about some of the recent advances that has happened. So this is one of them. So after 50 to 60 years of no FDA approved drugs for this disease, one of the first ones to get FDA approval in 2019 was a drug that's known as Oxybrita or Voxelotor, or the code name GBT440, which is a very interesting drug. This drug actually reduces hemoglobin's oxygen delivery capability. It actually increases the bond between the hemoglobin and the oxygen so that the hemoglobin is not able to release oxygen as efficiently. And then this actually helps hemoglobin in the sickle red blood cells maintain oxygen, and hence, and by doing that, it would reduce the polymerization. So this is a drug that was approved in 2019, as I said, but there were some concerns about this drug because you are effectively decreasing the oxygen delivery capacity of the blood and the red blood cells. So these patients are already suffering from low oxygen due to inefficient hemoglobin. If you further reduce the oxygen releasing capacity, then what's going to happen? So clinical trials mainly looked into that, and there are still ongoing studies looking at the rheological effects of this drug and also the oxygenation in tissues and so on. So that's one of the important drugs that are approved right now. And what becomes important in these emerging, these are also known as targeted therapies. It's quite important to monitor the patient's response, oxygenation, tissue oxygenation, blood rheology. One of the important outcomes of this therapy was increased red blood cell count or increased hemoglobin level in blood, which might increase blood viscosity. So these were all the things that we were interested in looking at with the red blood cell or blood rheology perspective. Another targeted therapy that was recently approved is by Novartis. It's called, the drug is called Adaq or crizanlizumab. It's essentially a P-selectin inhibitor. It's an antibody, it's a synthetic antibody that basically goes and binds to the P-selectin that might be exposed or that might be expressed on the endothelial surface if you have unhealthy red blood cells. So the previous drug was focused on the hemoglobin inside the red blood cells or targeting the hemoglobin inside red blood cells. This drug targets the unhealthy endothelial surface. As you can see, these drugs actually address different components of this microcirculatory problem. One is the hemoglobin in the red blood cell, and this one is the endothelial surface. There are other drugs that target the membrane integrity of the red blood cells or white blood cells in this complex scenario. So crizanlizumab is an inhibitor or anti-adhesion target for the endothelial surface. And in this case as well, one of the concerns is that P-selectin is typically utilized by neutrophils to adhere to endothelial surface and address issues if there are issues. So that's an adhesion molecule that exists for a reason, and inhibiting that might actually reduce or alter the neutrophil health or neutrophil function. So that's one of the reasons it is important again to monitor this type of treatment with companion diagnostic systems, and we have efforts in developing companion diagnostic like approaches for both of these emerging therapies in our lab. So another exciting development is the global emphasis or global interest in making these treatments more available to the patients who need them the most. If you remember, 99% of the people living with sickle cell disease live outside US, in Africa mainly. So NIH and Gates Foundation have been quite interested in making these treatments more available. And A good example is a private-public partnership between Novartis and the government of Ghana to provide everyone who is diagnosed with this disease an affordable treatment. Hydroxyurea is a drug that changes the hemoglobin composition in red blood cells, and it has been available for many years. It's a generic drug. It is cheap to produce. It can even be produced in certain countries in Africa. Novartis actually signed an agreement to basically provide the drug at a discounted rate to everyone in Ghana who's diagnosed with the disease. But the bottleneck has been, in that case, is the diagnostics, because there's no diagnostics. If you can make the treatment available at a low cost, but if you don't know who has the disease, or if you don't have a screening program, a national screening program, then that still becomes a bottleneck. And we have some collaborations with Novartis and Ghana to make the diagnostics more available through the new point of care systems that we have. Another interesting development is actually gene-based cures for sickle cell disease as well as HIV. If you are following the recent developments in gene therapies, there's an interesting observation that you can actually modify a certain gene in the T cells, and that might actually make someone immune to HIV. So this was actually observed in a condition in a patient in Europe, and from that the idea emerged, can we use gene therapy to cure or basically prevent HIV? Also, a similar type of gene therapy might also help with sickle cell disease. If you modify the hemoglobin, red blood cell producing stem cells, the gene in those cells, then you might actually start producing healthy version of the hemoglobin or non-sickle hemoglobin, or you can actually maybe bring the patient somewhere close to a trait, depending on the efficiency of the gene therapy. I will also talk about that. So Cure Sickle Cell Initiative, which I'm going to talk about a little bit later, it has been focusing on, this is a new initiative by NIH focusing on making cures available for patients with sickle cell disease. And this new joint venture between Gates and NIH focuses both on HIV and sickle cell disease, and they are focused on making these cures available to a broader public. So let me briefly talk about hemoglobin a little bit, because some of the concepts that I will talk about later relate to this. So hemoglobin is a protein that has 4 domains, alpha and beta, 2 alpha domains and 2 beta domains. One of the interesting things about hemoglobin is before we are born, before birth, we actually have this type of hemoglobin known as fetal hemoglobin. It's a fetal version of the hemoglobin that is inside the red blood cells, and fetal hemoglobin is made of different type of combination of proteins. It is in that case gamma and alpha chains exist. After birth, the gamma chain is replaced with beta chain, and the mutations, these mutations that I talked about, they all exist in the beta chain or the beta domain of the hemoglobin. There's no mutation that has been identified in the gamma chain or the gamma domain of the hemoglobin. So one of the approaches in gene therapy is to switch the production from beta to gamma, so that you can actually take advantage of the protected fetal hemoglobin that is mutation free. So you might be wondering, why we have a different type of hemoglobin before we are born. Why does this switching happen after birth, which is actually quite interesting, and it is typically explained by the oxygen affinity of these hemoglobins. If you think about it, before you are born, the baby is inside someone else's body, and the baby actually accesses oxygen through mother's blood. You don't really need that efficient of an hemoglobin. Now you actually need an hemoglobin that is more efficient in releasing oxygen rather than capturing oxygen from the environment. So the switching happens. So those problems, those mutations become amplified and become important after the switch happens. So this actually has many different implications. One is that the diagnosis and monitoring can start a little bit later. As you can see here, the gamma beta switch typically happens in the first few months of life. So you have a few months to screen all these babies after birth, and then put them on a certain treatment if the mutation exists. So if you also test these babies very early, like if you test them the first day after birth, then this will also cause some issues from the diagnostic perspective, because now if you have a high level of gamma hemoglobin or the fetal hemoglobin in the blood, it will make difficult to detect the mutation in the beta chain of the hemoglobin, because that's still in low concentration. So there are some implications for diagnostics and also for health-related reasons. So one of the things that was observed early on is that if you have a mixture of hemoglobin in your red blood cells, which is quite common, we all have different types of hemoglobins in our blood. But if you, for example, if you don't have a mutation, then you have a different combination of hemoglobins in the red blood cells. But if you have a sickle mutation, let's say, and if you happen to have more fetal hemoglobin in your blood cells, which might happen in the first few months of life, or there's actually an interesting mutation, a double mutation almost, that gives you a higher fetal hemoglobin after birth. So it's called high hemoglobin F persistency. So if you have that mutation in addition to sickle mutation, then you would end up with red blood cells that contain high levels of S and relatively high levels of fetal hemoglobin. And fetal hemoglobin has a protective role. As I said, fetal hemoglobin has doesn't really have any mutations that negatively impact the hemoglobin. So that actually protects the red blood cells from the negative effects or the impacts of the mutated hemoglobin. So if you have a certain percentage of fetal hemoglobin in your red blood cells, then that's good. And one of the curative therapies in sickle cell disease actually aims to achieve that. So they aim to disable that switch that happens so that you can actually end up with a mixture of fetal hemoglobin and adult hemoglobin in your red blood cells, which is quite interesting. So another drug, that the hydroxyurea drug that I mentioned, also does something similar to this. It increases the fetal hemoglobin to about 8 to 9%, which has beneficial effects. So one of the research directions in our lab is to understand the differences between red blood cells that have different composition of hemoglobin inside them, and also develop methods to quantify these levels of hemoglobin or composition of hemoglobin in red blood cells, which is actually not easy, surprisingly. So this is the switch that I was talking about. So if you can actually go and turn off the switch that causes this adult to fetal hemoglobin transformation, then you can actually have a beneficial effect on the patient. If you have a combination of or a mixture of fetal and adult hemoglobin, then you would go from a more severe phenotype to a mild or asymptomatic phenotype. This is kind of a trick or a cheating to a certain extent. If you think about it, you're actually turning off the switch to produce adult hemoglobin, and you're actually producing a combination of adult and fetal that can be a cure. One of the curative therapy approaches that is currently in the market right now or in the clinical trials right now is focusing on this aspect. So these are actually discussed in a recent review by Jennifer Doudna, who you might be familiar with the name. She actually talked about these different types of approaches in curing sickle cell disease in a review article early this year. So this is the genetic fundamentals of how the sickle cell disease happens. And on the right-hand side, there are two types of editing approaches or curative approaches are introduced. One is, the obvious one is fixing the mutation from A to 2 to T to A. So you can actually, if you can go and cut that mutated portion of the gene and then replace it with the correct one, which is a two-step process if you think about it, and you would have healthy red blood cell production. The other one, easier one, is to identify the gene that causes that switch from fetal to adult, and then go and disable that gene so that the switching does not happen. And then some portion of the hematopoietic stem cells in bone marrow still produce fetal hemoglobin containing red blood cells that would actually be potentially be a cure. If you can actually turn off this gene in 1/2 of your genome, then you would produce sickle and fetal containing red blood cells that would basically consider that would also be considered a cure as well. So different methods are in clinical trials right now, and one of the main research programs in our group is to help these curative therapy trials assess the quality of the red blood cells, assess the properties and the quality of the red blood cells after a gene-based therapy, which I will briefly talk about. So this is actually the approach that I was talking about. You start with hematopoietic stem cells. This is typically similar to very similar to bone marrow transplantation, if you are familiar with that. You take out the hemoglobin hematopoietic stem cells from the bone marrow. You use these editing approaches to either delete the BCL11A, or you do the correction in the gene and you put them back into the bone marrow, and then hope that some engraftment will happen. Some of these cells will go and home in bone marrow and start producing these healthy red blood cells. But the question becomes after this in the patients, what is the percentage of these hemoglobin A, F, S, sickle, fetal, adult hemoglobin, or hemoglobin A? And the other one is basically how to assess the percentage of edited cells, efficacy, quality control, and safety, and so on. So some of these clinical trials have been published in New England Journal of Medicine recently. There are still very small sample sizes for these. Only a few patients have been treated with each gene therapy in different parts of the world, but these are typically really impactful studies, and gene therapy in sickle cell disease has been considered as the poster child for the upcoming curative therapies and gene therapies, because if we can fix this single mutation, there's a good indication that actually we might be able to expand them to other diseases as well. So this is what we do in the context of curative therapy. So we help the Cure Sickle Cell Initiative at NHLVI assess the red blood cells after a patient receives a curative therapy. So we look at 4 fundamental properties: adhesion properties of red blood cells, or abnormal adhesion properties of red blood cells in the presence of other blood cells. We assess their deformability, how well they can clear through small openings in capillary mimicking channels. Another property that we look at is the density. So red blood cell density has been associated with the hemoglobin content. So we try to understand if the density has improved, and sickle cells have been typically associated with higher density. So we look at density for that purpose. And we also want to assess the composition of hemoglobin in these cells. So we want to be able to tell for every single red blood cell in a patient's blood after they receive a curative therapy, what portion of the what portion of the red blood cell is A, S, F, and so on. Another property that we'll look at is blood rheology, or viscosity in microscales. So that's another assay that we do. These are typically microfluidic assays that tell you that measure these properties in a standardized, reproducible, repeatable way as a quality measure after a gene therapy happens. I'm not going to continue talking about these assays, which are extremely exciting. Maybe another time I can talk about them. I would like to, in my remaining time, I would like to focus on the first things first, the detection, diagnosis. So here, as you can see, those gene therapies, looking at red blood cell properties, efficiency of gene therapies, these are the Western world problems that we have in sickle cell disease. those are good problems to have, but they typically currently relate to the 1%, less than 1% of the patients living with sickle cell disease. For that 99%, simple diagnostics is still the issue. They don't have access to diagnostics. So I'm going to briefly talk about our efforts in that area, expanding the diagnostics so that these patients can be identified, maybe kept alive with existing treatments, and maybe in our lifetimes, if these curative therapies become available, these patients can benefit from these curative therapies if they become available and accessible in the near term. So the diagnostics is typically done using electrophoresis. It's a method I'm sure you are all familiar with. It's in the WHO, World Health Organization, essential list of diagnostics for sickle cell disease. And anemia and hemoglobin level is typically analyzed using a system called hematology analyzer. That's also in the WHO essential list of diagnostics. So the challenges associated with even with these simple tests are cost, lab infrastructure, trained personnel, result interpretation, data management. These are the challenges. So what our approach was for improving diagnostics in sickle cell disease is to start with a method that's already well recognized, FDA approved, WHO recognized, and make it more available. So we start with electrophoresis in this case. We didn't invent anything new, fundamentally new. We made electrophoresis more available in this case. So our efforts in that case was to take the simplest version of electrophoresis, which is WHO recognized, as I said, and it is the most common electrophoresis method outside US in low resource settings, which is the cellulose acetate paper-based hemoglobin electrophoresis, which typically, the setup typically looks like this. It's a bench top-based system. So we took this system and made it portable with a single-use cartridge, much more faster, and it can be done by anyone, anywhere, without any additional, complexities. So one of the things that we also did was there was a step in post-assay staining step in the standard protocol. So we removed that step and replaced it with a high-resolution imaging that would basically image the hemoglobin bands after they form in electrophoresis, which again reduced the time and the cost and so on. So the video here shows you by the way, I just see a message here saying, please switch the screens, impossible to see the slides. Is there a problem? I'm sorry, I'm just seeing this message here. Can you see my screen or the slides?
I can see the screen.
Okay, do you see the slides?
Yeah, I see the slide.
Okay, sorry about that. Maybe it's fixed. So at the end, in electrophoresis, the typical way of running electrophoresis is actually you have different lanes, as you can see here, and the hemoglobin band that you put in this electrical field separates based on charge. Different hemoglobin types have different charges. A is the fastest moving hemoglobin in electric field, and different types of hemoglobins show as different bands here. Then after that, it becomes almost like a logic problem, the presence and absence of bands in certain locations to identify the mutations. For example, if you have a baby with a sickle mutation, this is what you would see. The second lane from the left, you would have some fetal hemoglobin and some hemoglobin S. That fetal hemoglobin disappears when this kid gets older, but then based on looking at these bands, you can identify. So we went from this lab bench based setup to a chip-based device that actually separates the hemoglobins based on their charge, and you can actually see different bands at different locations. And there's an algorithm that identifies the locations and relative intensities of these bands and basically presents the results to the user. So the user doesn't really need to do any interpretation of the results. It's done automatically by the device. So we also integrated total hemoglobin quantification to the same test. And by mixing the blood sample with the standard calibrator and separating the total hemoglobin from the standard calibrator in less than 2 minutes, and then looking at the relative ratios, which would give you an idea about the total hemoglobin level in blood And then if you wait for another 8 minutes in this electric field, then you would see the separation of the hemoglobin bands. From that, you can actually make the diagnostics. The good thing about this system is you can also use this system to monitor the treatments. If you have a gene modifying treatment, or if you have a fetal hemoglobin modifying treatment, then you would see an increase, for example, in the fetal hemoglobin expression, which can be monitored or quantified using this system. One of the key inventions, or one of the key IPs that we generated throughout this process, was to design and manufacture a single-use cartridge that would house the paper, electrodes, the buffer, the whole contents of the electrophoresis system in a single disposable cartridge. And we were able to manufacture these cartridges at high volumes, at low cost, and with Hemex commercializing this technology, now these cartridges are being produced in India and exported from India to other parts of the world at really affordable cost levels. So the engineering that went into this system was quite complex, but I'm not going to talk about that. If you have interest in how we designed this single-use cartridge, I would be able to talk about it, but there was quite a bit of heat and mass transport considerations that went into the design. Putting electrophoresis into a small cartridge, into a small plastic cartridge, posed many different problems that we had to engineer around heating, condensation, and the electrochemical processes that went into that and so on, but I'm not going to bore you with these details. So the timeline of this process has been relatively short. Actually, in 2016, the technology was licensed, and we had the first university prototypes for field tests, as you can see in the picture on the left. And then Hemex Health took over, designed the product, and released the product actually early this year in June of 2020. So now the device has GPS geolocation, Wi-Fi, Bluetooth touchscreen. It's battery operated, low cost. It's low cost and portable, and it can be done. The test can be performed by anyone, anywhere in the world. So this technology was clinically validated, as I said earlier, in different parts of the world. As you can see here, to account for the different types of hemoglobin variants that are prevalent in different regions, mainly focusing on sickle cell, thalassemias, hemoglobin E, and we have some ongoing tests around newborn screening in different parts of the world. And we have published these results over the years. I would be happy to provide the publications if you're interested. So this is what the user sees. They see the type of the bands that are present in the blood sample, and also the percentage. So for example, if you have a sickle cell trait sample, you would typically see 40-60 ratio of A and S, and the user would see a likely sickle cell trait result. Phenotype is identified as AS. Certain treatments might affect these results, so there's a note that is shown as transfusion can impact results, and you can see the other examples of result screens on this device. I'm not going to go into that. So these results are published now in the Analyst Journal. We tested all these different types of combinations of hemoglobin types in different parts of the world, in US, Nigeria, India, Thailand, and the results were pretty good, high accuracy. This slide actually shows the hemoglobin quantification potential. So this is the first and only point of care test that provides hemoglobin level, or the anemia state of the patient, and the hemoglobin variance. So you can actually have, in 8 minutes, you can have the results on anemia state, and also the hemoglobin variance in patients' samples. So that was quite useful in many regions. So these anemia or hemoglobin quantification potential was assessed according to the CLSI guidelines. Results are published as shown here. It shows pretty good accuracy and correlation with the standard method, which is the complete blood count, the hematology analyzer. And this is a quantitative test for anemia, so that's pretty good in that regard. So these are some of the results for anemia, non-anemia detection, the high accuracy, sensitivity, specificity that are much better than the existing point of care tests. So I don't have time to talk about it, but on the same platform, we can also do a malaria detection. So the malaria technology was also developed at Case, licensed by Hemex. And now on the same platform, you can perform anemia, hemoglobin variant analysis, and malaria. So this is a single platform that would address the needs of billions of people in the world where malaria and sickle cell disease or hemoglobin variants are prevalent. And Bio-Rad has been our distribution partner since the launch. So Bio-Rad is distributing these point of care devices now in India and in some parts of Africa. And as I said, regulatory approvals have been achieved in many different countries, CE mark. So it's, outside US, this technology is available, and we are now working on US FDA 510K approval so that this can also become available in the US. So clinical validation studies for the integrated system has also been done, sickle cell, malaria, and hemoglobin E. And you can see the results here. These results were presented in recent conferences before the pandemic, the in-person conferences. I only have a few minutes left, but I want to emphasize the importance of data. These technologies, these point of care technologies, at the end of the day, produce lots of data. These sickle cell clinics and these hospitals outside US in Africa already have challenges in storing patient data. You can see the patient folders on the shelves here, and typically they run out of shelf space and they give these folders to the patients. Data management is an issue. So we recognized this early on, and we wanted to have a plan for the data that will be generated by these diagnostic tests. And for that, actually, we partnered with an organization in the northern part of Nigeria, in Kano. It's a polio endemic region, and they actually developed these data management systems to track patients, to track, store the results, track the patients who have polio, and track the vaccination state of polio. So we work with an organization that has experience in these regions for tracking and management of data, and this is eHealth Africa organization, a nonprofit organization that is focused on electronic health records and data management in the low resource regions. And they have shown that with better management of data, you can actually improve for example, vaccination coverage. And we wanted to do something similar for sickle cell disease. At the end of the day, we need to basically monitor, or we need to track who has been screened, who has not been screened, and we have to have a better management strategy for the data. For that, we partnered with eHealth, and we received a grant from Vodafone Americas Foundation to develop an integrated point of care solution for this. So we have an app and a cloud-based system to store the data. These devices are all connected to Wi-Fi or the cell phone networks if it is available. And all the data that is produced in these point of care devices are uploaded to the database and made available to the patients and also the caregivers. So this is the overview of that system. So we have unique strategies for capturing the patient information, uniquely identifying the patient using biometric tools, and storing the data and uploading them and making it available to healthcare administrations, patients, and caregivers. So this is basically the back end of the point of care device. What are you going to do with the data? So that's an effort towards that. That's a still ongoing project supported by NIH, Fogarty, and Vodafone, as I said earlier. So this brings me to the end of my presentation. I hope I didn't go over time, but I would like to acknowledge the students in my lab who did all the work that I presented today, and the funding sources. And I would like to also thank you for your attention, and I would be happy to answer any questions if you have them. Thank you.
Thanks for the great talk, Umut. We had several questions in the chat box.
Yes.
One is, on what basis is cardiovascular disease least severe in that pyramid? You had the pyramid in the front.
Yeah, so I didn't intend to say that cardiovascular diseases are the least severe, but compared to those other diseases, the associations between cardiovascular disease and red blood cell health have been shown to be less severe. But there is actually more and more evidence that is coming out in the literature showing that the cardiovascular disease should maybe move up in that pyramid. The severity, the association with red blood cell health and microcirculatory health is being recognized more and more every day. So that's a pyramid that I actually made myself, to be honest. It's not published anywhere, so the order might be flexible or fluid and it might change over time. But I meant to basically put it at the bottom of the pyramid because it impacts the most people in the world, and the associations have been shown to be less clear, maybe compared to the other ones. But it's a good question.
Will heterozygous allele also result in the disease?
Sorry, could you please?
Will heterozygous allele also result in the disease?
The heterozygous AS is, there is actually a big debate in that area. Many people, many physicians and many people actually, many researchers think that AS, the heterozygous state, is like almost like healthy, but there are certain indications that heterozygous state might be associated with kidney disease. So for example, people who have the trait, sickle cell trait, are more likely to have kidney diseases. So there is a link, there's a potential link between the microcirculation in kidney and the heterozygous state. So there are some indications, but the links are not very clear yet. But the trait, understanding heterozygous is extremely important in the context of curative therapies, because the expectation is that many of these curative therapies will create patient phenotypes that will be similar to a trait. Since gene therapies cannot typically work with 100% efficiency, if you correct a portion of the cells in bone marrow, then these patients' bone marrow will start producing cells that are probably half A, half S, or some A and some S. So that's understanding the trait, understanding heterozygous phenotype has been extremely important more recently with the emergence of gene therapies, but it's a very good question too.
Another one is, you have a recently published patent about dielectric sensing for sample characterization.
Mm-hmm.
Is it possible to use this system as a patch for applications like continuous monitoring of blood glucose in diabetic patients? If yes, how it can be performed?
So we haven't really looked at the blood glucose applications or diabetes applications. That recent patent is mainly focused on monitoring blood coagulation or thrombotic effects of these issues. For example, if you have a blood coagulation disorder, hemostatic disorder, by looking at the dielectric spectroscopy of blood, we think that we may be able to tell if the blood clotting process is different than how it should be, and how certain, for example, oral anticoagulants might affect or improve that coagulation process. So it's essentially a bulk measurement tool to look at blood coagulation as it happens real time in these devices. So one of the companies, the Exatec company, is actually running clinical trials right now in looking at the dielectric spectroscopy of blood to diagnose and monitor coagulation disorders, but we haven't done any work in diabetes and blood glucose space.
Okay. In your experience, what's the best way to assess the quality of a red blood cell? Second part, what is the best non-biochemical method of assessing this without lysing the cell?
Yeah, so most of our research in our lab is actually focused exactly on this question. So we keep the cell live and intact, and we look at its rheological properties, which is quite, there are many actually. We think that adhesion, deformability, and microscale rheology, microscale viscosity are key properties in defining the functional quality of red blood cell. And for example, a healthy red blood cell should never stick to a surface. We expect red blood cells to freely flow in the circulation. But when it comes to in contact with endothelial proteins, if a red blood cell sticks to endothelial proteins, then that's a bad sign. We don't want that. So that's why we use that as one of the quality assessment tools. Deformability is another one. The red blood cell should be able to pass through small openings, 2-3 micrometer size openings without any issues. So we test how well they can pass through these small openings and whether or not they cause occlusions. One of the unique approaches that we have in our essays, which is becoming more and more important more recently, is how do these properties change in the case of hypoxia? So red blood cells are designed to perform as good in normoxic conditions, for example, in the lung, versus the most hypoxic regions in the body, such as the kidneys. So you should have the same biophysical properties ideally in these cells in both normoxic and hypoxic states. So we have custom assays to look at, for example, how well a red blood cell performs in terms of these key capabilities in hypoxic and normoxic states. In certain cases, hypoxic properties are much worse in certain diseases, and that becomes like an important biomarker, a biophysical biomarker for red blood cell.
Okay. Final one is, we know first human embryonic gene editing was on a hypertrophic cardiomyopathy, a dominant heterozygous disease on 2017. What's the challenge to do the same procedure for sickle in zygote for single gamete in pronucleus to treat disease and protect trait situation?
So that's a good question. I have to say that I'm not an expert in gene editing, so we help assess the red blood cells that emerge from a gene-edited bone marrow, for example. But in the case of sickle cell disease, I think maybe a good way to answer this question is bone marrow transplant to cure sickle cell disease has been around, has been one of the most available, or it's not available yet, that widespread yet, but it has been one of the accessible versions of curing sickle cell disease. So bone marrow transplantation, allogenic bone marrow transplantation has been done for many years now. So the difference in the gene editing is taking patients' own bone marrow instead of a donor's bone marrow, for example, in that case, taking patients' own bone marrow, in vitro editing it, and then transplanting it back to the patient. So it doesn't really involve an embryo, as you can imagine here. So this involves patient's own consent, patient's own bone marrow. And I think the key bottleneck here is the efficiency in editing of these hematopoietic stem cells outside the body. So the Gates NIH efforts in that regard is focused on in vivo editing. So can you, for example, maybe related to some of the work that you do at Terasaki, can we improve better ways of delivering these gene editing tools to the bone marrow, hematopoietic stem cells in bone marrow, without the need for taking the bone marrow cells outside the body? The expectation is that in low resource settings, the most feasible solution will be these in vivo gene editing tools. It's extremely expensive and extremely resource intensive to do this. Take the red blood, take the hematopoietic stem cells outside the body, edit, put them back. That's a long process, very expensive process. So I think the drug delivery, gene delivery will have a huge impact in this area if we can achieve these single mutation editing approaches in vivo with better delivery tools, with improved delivery tools.
We should probably set up a second meeting about those topics.
Yes.
A couple other ones. Will heterozygous, okay, we asked that one. Effect of Oxybrita on oxygen delivery capacity of hemoglobin was a bit unclear.
Yes. So the Oxybrita, I think all these therapies deserve a presentation on their own, to be honest. But Oxybrita is quite interesting. It changes the hemoglobin's affinity to oxygen. So it prevents hemoglobin from releasing oxygen when for example, red blood cells travel to a low oxygen place in the body. So it prevents the oxygen dissociation. It changes essentially the oxygen hemoglobin association, kinetics. It makes the oxygen more difficult to release from the hemoglobin. This is an effort towards preventing polymerization of sickle hemoglobin in hypoxic states. So hemoglobin polymerization happens in low oxygen conditions or when the hemoglobin releases the oxygen. So if you keep the hemoglobin stuck to the oxygen, it cannot polymerize. So that's how that drug operates. But the concern is that if you are reducing the oxygen delivery potential of the hemoglobin, then are you helping the patient or not helping the patient? You know, that's not clear. Overall, clinical trials show to be positive because this drug also have been shown to increase the hemoglobin level in blood and red blood cell count in blood. So lower hemoglobin dissociation has been compensated by increased levels of hemoglobin levels, increased hemoglobin levels and increased red blood cell counts in these patients. That has other implications, that is other rheological implications, which we can maybe talk about some other time. But, you know, I hope this answers the question for now.
If another one is gamma chains reduced in adult cells. How does one switch from beta to gamma again?
So there is a BCL11A gene that is responsible for making that switch after a baby is born. BCL11A, if you go and delete the BCL11A gene, make a note of BCL11A and read. It's very interesting. It's a very interesting gene that does that switch. So if you delete that BCL11A gene with using gene editing tools, then that switch cannot happen. So the body is not able to switch from gamma to the beta, and then the body keeps producing the fetal hemoglobin instead of adult hemoglobin, which is quite interesting if you think about it. But even that gene deletion is not highly efficient. You take out, again, stem cells from the body. You cannot delete that gene in 100% of the cells. You delete it maybe in a portion of them. And in certain cases, heterozygously. In a single cell, you might delete one of the genes, but not the other. So this cell might produce half A, half F. And it's important to know what that percentage is. And the engraftment efficiency is also low in these cases, as you can imagine. So the first efficiency is with gene editing. The second inefficiency is with the engraftment, which might result in like 5 to 10% corrected or deleted genes in these patients. And, you know, our work as engineers in that area is, for example, looking at the rheological properties of blood after a person receives gene therapy and figuring out what percentage of efficiency is sufficient. You know, at what level you see an insignificant amount of adhered red blood cells or sticky red blood cells or non-deformable red blood cells, you know, what percentage of engraftment or efficiency would result in a significant improvement that can be considered as a cure. So this is the definition that we are working on right now with these new microfluidic tools.
For malaria, how does the technology deal with hemozoin sequestration during stages of the disease? Essentially, hemozoins disappear during some of the stages of the disease.
So that's a very good question. You know, I get this question all the time. We have done actually some work in that. I'm not a malaria expert, so to say. I'm helping the team with engineering expertise. But this question is a very good one. Detecting hemozoin in blood has been quite challenging for many years. And some of the key innovations that Hemex did in the hemozoin detection area was on efficient isolation of hemozoin from blood, which involves efficient lysing of red blood cells, efficient lysing of the parasite and the vesicles inside the parasite. So that information that is asked in this question, you know, that hemozoin disappearing at certain stages was because the current methods or that existing methods was not able to detect the hemozoin levels in blood in those stages. So we actually did studies, clinical studies in different parts of the world, showing that hemozoin is always present in blood if you have the right detection tools. I would be happy to share those papers or publications or information if you are interested. And we recently published a paper, Hemex actually published a paper in eClinical Medicine, showing the first proof of concept or first clinical proof of diagnosing malaria based on hemozoin in blood. So that has not been an issue for the detection system that Hemex has developed. Maybe this is the short answer.
Here's another question. Considering similarities between corona and sickle pathophysiology, like vasofatigue, vasoocclusion, HB desaturation, hypoxia, is there any treatment that you think would be useful for corona?
Yes, actually, that's a very good question. Actually, we submitted a number of supplemental grants to the COVID-19 opportunities at NIH. We still haven't heard about the results yet, but some of the drugs, for example, some of the drugs that I listed, crizanlizumab, for example, P-selectin inhibitor, might be a good drug candidate for helping with especially the lung microcirculation related issues, you know, the thromboinflammation in the lung microcirculation. That P-selectin inhibitor is a good candidate. Actually, we proposed to study that in a lung microcirculation model in one of our applications. Another one is a red cell targeting drug, imatinib, that we published recently on in British Journal of Hematology. Imatinib is actually in the list of potential therapeutics for COVID-19. If you look at that list, that actually changes red blood cell properties and also helps with the, I think, I forgot the mechanism exactly right now, but there's a proposed mechanism for the coronavirus infection of the cells. But yes, so there are lots of similarities between sickle cell disease symptoms and COVID-19. In fact, patients with sickle cell disease and trait are a high risk group for COVID-19. So their survival if they get COVID-19 is pretty bad, actually. You know, the survival rates are pretty bad. So that's why these people living with microcirculatory disorders need a special attention when it comes to COVID-19. That's a very good question, a very good observation.
Well, thank you for this great talk. Appreciate you joining our seminar. Come visit us soon.
Thank you, Mehmet. I just want to mention I just want to quickly mention that, you know, you have seen the type of work that we do, especially in the case of in vivo gene editing. If you have any interest in talking about anything related to that, I would be happy to maybe schedule another call, and I would be also happy to give another talk if you are interested, specifically focus on what we are doing with gene editing, you know, what type of assessment tools we are developing, what are some of the challenges that we face, and so on.
Yeah, let me talk to the group and get back to you. Thanks again.
Yes, thank you.
Bye-bye.
Bye.